Today, we are taking a look at food tests, under which we are considering identification of food nutrients. Carbohydrates. Iodine test: cut a section of carbohydrate food to expose the flesh. Then pour drops of iodine on the exposed flesh. If it turns black or darkish brown, it shows the presence of carbohydrate. If it remains light brown, then carbohydrate is absent. Litmus test: this is another simple experiment to detect the presence of carbohydrate in any food substance. The food item is peeled. If it has any bark, it is then grated and made into a paste. Red litmus paper is immersed into the paste. If it turns blue, it shows the presence of carbohydrate, but if it remains unchanged, it does not contain any carbohydrate. Proteins. Foam test: a very simple test for the presence of protein, is to dissolve the food substance in water and shake vigorously. If a foam is produced, it shows the presence of protein. Absence of foam indicates that the food substance does not contain protein. Alcohol test: alcohols have a coagulating effect on proteins. Thus, if a food product or its solution coagulates, shrinks, or curdles when immersed in alcohol, it shows that food is proteinous. In the case of meat, apart from shrinking, its color also changes to brown. Millons test: Millons reagent may be used to detect the presence of protein in foodstuffs. A solution of the foodstuff is warmed with Millons reagent, which contains mercurous and mercuric nitrates in nitric acid. If protein is present, a white precipitate which later turns red is obtained. The only common protein which does not give a positive result with this test is gelatin. Fats. Blotting paper test: the presence of fat in any foodstuff can be detected by grinding a small quantity of foodstuff between two pieces of blotting or filter paper. If after grinding, the paper or some portion on it becomes translucent or greasy, it shows the presence of fats or oil in the foodstuff. If not, it indicates the absence of fat.